So we are from the Carbon Capture Group, Transportation Group 2. And for our project, we wanted to focus on the only indigenous carbon capture technology to planet Earth, which is biological sequestration with biomass. Our project focused on a biomass called duckweed because it has many unique characteristics and advantages that make it a much better feedstock for carbon reduction technologies than the current feedstocks such as sugarcane, corn, and soy. One, duckweed grows extremely quickly, able to double its yield in under 48 hours in ideal conditions. And two, it's really interesting how it increases its yield because it does so on swine or other industrial wastewater. So it is able to grow and actually thrives in conditions that can kill most other plants. The third unique characteristic of duckweed is that it can be produced for either high starch or high protein content. This means that you can produce multiple co-products, which then through a feasibility study you can determine which product, which avenue of production, which type of duckweed has the most potential for carbon reduction. So in looking at these three unique characteristics, this led us to our case study area, Brazil. Brazil is the fourth largest swine producing region in the world and is the second largest producer of ethanol. So we saw a big possibility for integrated farming producing both duckweed and swine. And the main productivity for ethanol production in Brazil would occur in the south center-west region, which includes Paraná, São Paulo, and Minas Gerais. And this region already has existing infrastructure. There 76% of ethanol is produced in this region, so you already have the biorefineries in place. Also, 34% of swine is produced in this area. So we produced a theoretical yield for this region, the center-west region, and found that we could produce a significant amount of ethanol using academic reports of 6,500 liters of ethanol per hectare of land. More importantly, duckweed doesn't grow on land, it's a floating plant. So by growing duckweed and producing ethanol or other products, we reduce also the land impacts. We have no deforestation, we're not cutting down rainforest or cerrado land. We are growing this on existing wastewater swine facilities like waste stabilization ponds or constructed wetlands. Now the rest of Brazil, outside of the center-west region, we called this Zone P for zone protein because the potential for duckweed production is large. There's a lot of swine production, over 65%, outside of that region, but there's no infrastructure for biorefineries, so we can't refine ethanol. So what we do in this region instead is we substitute duckweed protein using high protein duckweed for soybeans. And what this allows us to do is reduce, as I said, the land impacts from soybean production, which are even more significant in the edge of the Amazon region and the Cerrado region in the north, central, east regions of Brazil. We're able to do this because the amino acid profile of duckweed protein is very similar to soy protein, and that led to the assumption that we can do a direct substitute. And to make this scalable to Norway and to make it interesting for Norwegian investors, we did a little feasibility study on Norwegian import of soy and also Norwegian ethanol production. At the current level of ethanol production in Norway, to meet the demands for 5% of gasoline being ethanol, we were able to save 25 kilotons of CO2. And also, if we were to directly substitute the amount of soy that we import to Norway for example, feed for the aquaculture industry or feed for pork and chicken production, we could reduce our CO2 emissions by 250 kilotons. If you put these two numbers together, the actual carbon emissions would be approximately 50% of Norway's 7th largest polluting factory, which is currently an aluminum production facility by Hydro. So the summary and conclusion of this project is that we believe our numbers are interesting, and we believe that further investing in this project and in this closed integrated farming system is worth a look.